Hello everyone. Can everybody see me here on the channel, on the YouTube channel? Just the first check, so I can see. Okay, it's live here. It's working. Just making some first checks here. Just the first check, so I can see. Okay, it's live here. It's working. Can everybody see my screen as well? Checks here. Just the first check, so I can see. Okay, it's live here. It's working. Can everybody see my screen as well? Can everybody see my screen here? I'm sharing the screen. Alguém consegue ver? Todo mundo consegue ver minha tela aqui? Okay. Yep, okay. So, I am Hernani Oliveira. I got my PhD from the University of London in the UK, working with molecular food webs and the effects of the strongest immune records on the molecular food webs of insectivorous and frugivorous bats in Costa Rica. And that's where the idea of this course started. So I'm going to talk about a little bit about how to integrate molecular data into ecological networks, some perspectives and challenges. I'm now a young talent research fellow in the Universidade Federal do Parana under the supervision of Fabrício Domingos, also from the same university. And that's it. And so let's go to start talking a little bit about molecular data and ecological networks. So to start with, to analyze and to understand interactions and species interactions is a very hard and difficult thing to do, mainly for two aspects of this challenge. First, because species are a very difficult thing to describe and to identify in the field. So we have a lot of species that are they're morphologically very similar, but they actually represent different taxa. And this is hard to sample when you only have, or you only use morphology to do that, or any visual clue to do some things like this. So you can see, for example, in this figure, how all these butterflies, or many of them, they look very similar in terms of the color, the shapes of the wing, the size, but they actually represent in many cases different species. And you cannot, or it's very difficult to see this difference in the field without sometimes analyzing the genitalia of them, or even some more microscopic aspects of the morphology of this species. So this is the first challenge. So when you think about ecological networks or interactions between species, you need to think first about the species. So you need actually to be able to identify these species in the field in order to study with whom they interact with. But we can see already that this is a big challenge because many of these taxa, they are very similar, and it's very hard to identify and to differentiate them in the field or even in the lab sometimes. On the other side, even if you identify the species, interactions are also very, very hard to sample as well. And some of them might take decades to be recorded, or even centuries. This is a very specific case of an orchid from Madagascar that has a very interesting story behind it. So Charles Darwin received this orchid when he was studying about the evolution of different species, and he saw the size of the nectar, the feeder of the orchid, and he saw that it was one of the largest ones that he has ever seen in his life, with more than 20 centimeters. And he predicted at the time, even though he had never seen this orchid in the wild, he predicted at the time that there should be a pollinator with probably a moth that should have a feeder that would, or a proboscis that would have the same length of the orchid in order to be able to pollinate it. But at the time, and even much later, nobody has ever recorded any moth with a proboscis this big, or even recorded this interaction in the wild. And he predicted this in 1862, and it was not only until 1992 that someone has actually recorded the interaction in the wild, this interaction in the wild. So it took more than 100 years for people to be actually able to record not only the species, but also the interaction to be happening in nature. And this is one of the most famous cases of predicted interactions that should be happening in the wild or in nature, but took a long time for people to actually be able to see this happening. So you can have an idea here about how difficult it can be, or how challenging it can be sometimes to record interactions, especially when they are rare, or when they don't occur so often in nature. And this poses a big dilemma or a challenge. That's the idea of the missing links versus forbidden links. So missing links are links that you haven't observed in your network, or you have never observed this interaction, but doesn't mean just because you have never observed an interaction in the wild doesn't mean that the interaction doesn't exist. And this interaction between the moth and the orchid is a very, very illustrative case of this. Sometimes the interaction can also be a forbidden link. What we call is that an interaction will never happen simply because both species never co-occur in the same place or at the same time. But when you have these rare interactions, it's very hard to tell sometimes if this is a missing link or is a forbidden link. So if this is not happening simply because you have never sampled enough, like all the interactions in order to be able to catch this interaction, or if it's a forbidden link, or it's something that you have never observed simply because they never actually occur in the wild for many different reasons, such as the case of the polar bears and the penguins. They will never be able to interact naturally in the wild simply because they don't co-occur at the same place at the same time in order to be able to have any kind of interaction. And this is interesting because when you look at the ecological networks, they are shaped by many different factors. So you have phenotypes of the species. So morphology can play a big role in terms of how two species will interact, which will ultimately lead to trait matching. So a moth, taking the example of the moth, a moth will only pollinate an orchid, or the orchid will only be pollinated by a moth that has a proboscis that's long enough to actually reach the nectar, in order to be able to feed and also to transfer the pollen to another orchid. But also a lot of different aspects of this, the structure of the networks of interactions that are shaped by many different factors. And one of the main ones that I'm going to talk about with you today in this talk is going to be related with sampling effects. So how do you actually increase your capacity to sample the interactions, and how do you deal with this trade-off as well? Because at the same time that you're able to sample more interactions, you have changes in the known structure of the networks that you will need to address in order to be able to analyze these networks. So I'm going to focus more specifically in terms of how you can apply molecular data and incorporate them into ecological networks in order to get more, to assess a higher definition of the interactions, and also add much more missing links that you would never be able to address them or to study them if you were not using molecular data. And which makes us also closer to come to a point where we're talking about the true network structure. So if you think about what we observe in the wild versus what the ecological networks really look like, there's a lot of these missing links. So when you use traditional data or traditional approach to characterize the interactions, some interactions you will never be able to actually observe in the wild. For example, when you study the feces of many different species, many of these feces, they have reminiscence of the prey items that they eat, but many times you cannot identify them at the species level or the more lower level, taxonomic level, just because you don't have enough evidence of any morphological trait that you can address them or you can place them in a species. So most of the species that are part of a diet of many different taxa, you'll never be able to describe them in the diet of these species using traditional methods, simply because these methods are not good enough to describe some of these interactions. So when you add molecular data, you catch much more of these missing links, and you become closer to observing a structure that what we would call like the true natural structure, or or how the links they actually happen in the wild versus how we're actually perceiving them. Just to think about a little bit, there are many different ways that you can sample interactions, and you can consider species interactions, many different kinds of things. Two people interacting as friends could be an interaction. The diet of species could be an interaction between the species and the prey items that it has on the diet. But I'm focusing more specifically here in the talk about dietary interactions. There are going to be other talks focusing on this as well, but I'm going to talk more specifically about how to incorporate the molecular data to give a better resolution of the networks, and also how to address some of the issues coming up from using molecular data into ecological networks. But you have many different ways to sample interactions. So you can use camera traps to actually record the frugivorous rates of mammals or different species in the wild. You can actually observe them as well. So you can sit in a rock and just observe which animals are coming to feed on that fruit, on that flower, or anything else that you're actually studying. You can actually also collect the animal in the wild. So you can, for example, catch a trout and then see what the trout is eating, which many people do. But you can also use molecular data or molecular methods in order to sample the DNA of these to describe these species and describe these interactions. So you are able to catch a lot of variation and interactions and links that you would never be able to find in any other of these other types of methods. This is an interesting study from Pringle and Hutchinson. Actually, they're showing the study that they added molecular methods to describe the interactions of some herbivores in Africa, in Gorongosa National Park. And they actually saw that by adding molecular methods, as we have talked before, they added much more links, and they considerably changed the structure of the network of interactions. So when you add molecular data, you're not adding just more information. You're actually changing the way that we perceive, or the structure of interactions that these networks actually have, or our understanding about how they actually are so far. And this goes through many different levels. So you have, there's an article that's mentioned in this article that they call the demons of resolution. So when you work with molecular data, one of the main challenges to work with molecular data is also which taxonomic level are you actually going to be describing this species. So you can be using general level, you can use family level, sometimes species level as well. But it's very common that we use motus to actually describe these species when we talk about DNA barcoding, which is also one of the main focus of the talk as well. What are motus? Are basically the way that we have created to describe the genetic diversity of a pool of sample of DNA in order to compile this genetic diversity in something that would be a proxy for a species. So actually called motu, but it would be something that we have equivalent, it's not the same, but it's equivalent to species in this kind of studies. But then just to make sure that you understand this part as well, it's important to frame that the resolution of the network is also important in order to understand the structure of the interactions. So if you focus on motus or species, you have a resolution that will go, you have a network with certain structure, but if your resolution of your network or interactions go to general level, you also have a different perception of these interactions. I'm going to talk about this later as well in more detail in the talk. And if you go to family level or a higher taxonomic level, when you're lumping or putting together interactions from different species, you also change a little bit the structure of the interactions of the network. This is extremely important because many times when we don't add molecular methods or DNA barcoding in this case, you are more restricted to higher taxonomic levels of the identification. So usually you go to family level, general level, but very rarely you can go to species level in many of the descriptions of the diets of different taxa. But when you go to use DNA barcoding, you can get a more specific description about these interactions and more detailed as well. I don't know if everybody's familiar with DNA barcoding, but it was an initiative that started in the beginning of the 2000s. And they have an initial proposal of being able to describe all the diversity of many different groups, animal groups, based on a single or few molecular markers that would have mainly three characteristics. So they should be have enough genetic variability and divergence between them, so you can separate and differentiate between different taxa. They should possess flanking sites for developing universal primers as well, so you need to be able to make primers for these molecular markers in order to recover them during these studies. And also they should be very short, so they should have only few hundreds or dozens of DNA pairs. So this is one of the main characteristics of this DNA barcoding. So they have created this idea, or started to develop this idea in the beginning of the 2000s, and then suddenly became a very big consortium, not suddenly, across time, very big consortium. Today, nowadays, is the biggest biodiversity consortium in the world, where every country has one group of researchers working with DNA barcoding and developing, describing species as well, using them to describe species, which has started, as I said, like a big, not only a consortium, but a big initiative worldwide. So all the countries now, they have their own facilities, and also most of them, they have their own facilities and all initiatives in order to describe their biodiversity based on this consortium. So we're describing biodiversity of fish, of mammals, of frogs, of everything that you can think about that can actually be described using mitochondrial DNA, basically, mostly, in many cases. And there's also one important thing in terms of the advance of the use of this technique, is that the cost of sequencing DNA has significantly decreased over time in many different places. Some places, there's still a lot of improvement to do on this, because it remains a little bit expensive still, especially depending on the currency, from the dollar, from the pound or the euro. But in general, the cost of sequencing DNA has considerably decreased over time, to a rate that now it's possible to use this technique to sequence DNA, a quantity of DNA that in 20 years ago it would be impossible, just because it would be too expensive to do something like that. But today the price has decreased so much that we can apply sequencing techniques, as Fabricius also does in his research as well, but also other people do in Brazil and other places as well, in order to apply to these studies and to use DNA and molecular methods in their own, to answer their own ecological questions. And this has triggered a big variation also in terms of the questions that they can answer and the approach that people use them. So you can use DNA barcoding or molecular methods to answer a range of different types of questions and a range of ecological context as well. So you can go to answer how ancient ecosystems have changed sometimes across time, how plant-pollinator interactions they actually occur, or describe the plant visitors of certain flower species just based on the DNA that is left on the plant or on the flower. You can describe the diets of many different species, so species that feed on insects, species that feed on plants, or many different types of taxa. You can detect sometimes even invasive species, so you can use this as an environmental DNA and sample parts of the environment, such as water courses, in order to see which species are present in these environments. You can see the response of species to pollution, and even you can catch now. It has been shown to be able to that we are actually able to catch EDNA from the air. So there's a range of different possibilities here that we can use DNA barcoding, especially molecular methods, in order to study not only interactions between species, but also use them to answer ecological questions in many different kinds of contexts worldwide. And this is very interesting because it has triggered a big surge in terms of the number of species that has been described in the last 20 years since this technique has been developed. So if you think about geometric species, for example, about Lepidoptera, since Linneo started to describe species, we have roughly 23,000 species of geometrids that have been described. But when you think about the late 90s and early 2000s, and especially since the early 2000s when this technique was developed, they used this to describe roughly 20,000 geometric species in just 12 to 20 years. So it's a very big increase in terms of our ability to actually not only describe interactions, but also to describe species. But this has the addition of also describing interactions as well. As we're talking about, we can use DNA barcoding not only to describe as a complementary method to describe species, but also to describe the species present in a place or in different places. So we can actually use DNA barcoding also to describe the species present in the feces of another species, for example. And this way you can describe the interactions. So the number of interactions that we have been able to sample after the development of DNA barcoding has also increased exponentially. And this, as I was saying before, has also changed the perspective of how the networks are actually structured and how these interactions actually happen. So if you think, for example, of these two rabbit species or the hare species, before DNA barcoding, they were considered the same species. So if they are considered the same species, you have a network of interactions where the orange dot represents just one species. But now that you have a technique that's powerful enough to actually distinguish them, you can actually insert this into the analysis, and then now you have not only one species with not only one node or in the network with two species, but now two nodes with different species. So actually changing the structure of the network and also our perception about how these interactions, they happen in the wild. And this is the same also for other very different taxa, such as Lepidoptera, for example, as well. Before these four species, they are morphologically very similar. They are clumped into just one species. But after the development of DNA barcoding, they have been described many other different species from one single complex of species. And now we have a different perception about how they actually interact, not only with the food plants that they eat as caterpillars, but also about which other species parasitoid them. So we have a massive increase in terms of the resolution and the perception about how the structure of these interactions actually are in the wild, and we're getting closer and closer to what would be the true structure of these networks instead of just how we perceive them. Because before there was a big biases in terms of which methods we're using and how good we are in terms of describing them. But this is very interesting improvement and development, because when you start adding more species in the networks, there are some metrics of the networks that they change, but they change differently if you have a network of many species interacting or few species interacting. So we're going from a place that we have just few species described because we're not good enough to describe them, to a place where we have a lot of species described and also a lot of interactions happening between these species. The interesting thing about this is that when you have a network with just few species interacting between them, any change in this interaction, any change in this network makes a considerable change in terms of the structure of the network. So if you add one species or add few links, because the interaction, the network or the matrix of interaction is so small, any addition or removal of species or interactions make a big difference in terms of the structure they're actually observing. But when you go to bigger networks, which is more our case now with the addition of DNA barcoding, where we're not able only to describe more species, but also to detect these interactions in the wild, we have much bigger networks. But this comes to with the price of another of a cost. So we have more species described and more better described networks of interactions, but because our network is becoming very big, it's much harder to actually sample this network good enough in order to know most of interactions present on these networks. But this is not because they were not there before. It's just because now we're much better in terms of perceiving them and describing them. When before they were also there, but we were not good enough in terms of detecting them and describing them. So we went from a scenario where we have few species described and interacting, where any new species described or any link described made a significant change in terms of the interactions, the structure of the network. But we had a very high sample completeness. So we thought we knew exactly how the network looked like, because we have sampled most of the species that we could, to a scenario where we had many more species described. And we have seen that the structure doesn't change so much if you add many more species instead of, in the case of smaller networks, but it's also much harder to sample most of the species present in the network. So the network metrics, many of them are robust to adding more species, so you can add more species and links in a big network using molecular methods, which usually doesn't change significantly the structure of the network, but it's very hard to sample these networks well enough. So to reach 90%, 95% of the interactions of the true network sampled is a much harder effort or a much harder challenge to do. And this is very well shown in this figure, where you can see that networks that are smaller, so on the left, they have a much higher sample completeness. So you know much better networks that are formed by few species and few interactions than networks that are formed with many species and many interactions, just because it's much harder to sample them. You have many more possibilities, and many of these possibilities can be rare as well, so you do not observe them very easily or very often across time and across space, which makes the networks very poorly sampled in terms of how much interactions actually are there. So we have this challenge in this dichotomy a little bit, because we're better at describing the interactions and the networks and the species that are present in these networks, but at the same time, because you have more species interacting, makes in much more possibilities of interactions, it makes much more difficult to actually sample these networks well enough. So it's a trade-off a little bit between adding more species versus, not a trade-off, but it's a dichotomy, it's a problem in terms of describing more species and understanding better the networks, but also you need to make a much bigger effort if you really want to know, if you really want to sample all the interactions in these networks and these species as well. But we're all ecologists, so we're used to deal with these problems in our studies. So there are some metrics that they are more robust to this kind of changes. So if you have a small network or a big network, doesn't make a big difference in terms of the perception of the structure of the network. So the network doesn't change so much in terms of the topology and the structure if you have a small network or a big one, for some of these metrics to measure the structure. So if you're measuring something that should be changing a lot across time or across space, and you're constantly adding more species, or you're comparing networks with different species, you can focus on some few network metrics that they don't suffer a lot of effects from the network size in order to compare these networks. And the good thing about the working with DNA is that there are DNA everywhere, basically everywhere. You can find samples of DNA. You have DNA in the wings or on the body of an insect that's transporting pollen from a flower to another. You have DNA on a fruit that has been eaten by a frugivore. You have DNA on the seeds that have been dispersed by the same frugivore. You have DNA on insects that are feeding on blood, for example, as well. So you can virtually find DNA anywhere. You can find even in the air. Like, this is a very recent article that has came out a few days ago from my supervisor from the PhD, where she has showed that you can actually even collect and describe DNA that has been collected in the air, present in a closed room. So we have a lot of potential applications of these methods. There's also a specific way that you can sample them, so you can use these methods and collect DNA from these different surface in order to describe interactions. So if the species is present there, they're going to leave DNA, and if you collect the DNA there, it's showing that the interaction is present between these two species. We also have a big depository of data. We have many of them, but one of the most famous ones is the boat systems, where we have currently more than 230,000 species that have been deposited on it. So you have a wide range of possibilities that you can match your sequence with a potential species that might be stored on the on the database. Sorry. And you can collect also DNA from different surfaces, as I said, which you can use to describe different types of interactions. So you have DNA that you can sample, for example, from flowers, where you can describe the visitors of the flowers, but you can also use this to look at the different perspectives. So you can look at the DNA present in the body of a pollinator to see which flowers this pollinator has been visiting as well, and use this information to describe the network of interactions between the pollinators and the plants, or the plants and the pollinators, depending on where your focus are. And in this case, we're able to make these big networks of many different groups. So you can make a network of pollinators, ectoparasites, prey, plants, which if you do this just with traditional methods, it would be mainly or very difficult or even impossible in some cases. And come closer to something that we call the true network structure of what's actually happening in the wild. This is a very interesting study that has triggered this kind of ideas as well, or has given more support for adding molecular methods to sample interactions, because they have used molecular methods to sample a food web in Greenland, so very close to the Arctic, where there's very few species, so supposedly very few interactions happening between the species in the food web of parasitoids and hosts. And even after many years sampling this network, when they added molecular methods, they were still able to describe new interactions. So it's a very simple system, very few species, and when they added molecular methods, they have found species that they haven't detected before. So you can see that not only you can find more interactions, but also the structure of the network also changes when you add molecular data to describe these interactions and the structure of these networks, what is being shown here. I'm going to focus a little bit more about studies on bats, which is one of the main groups that I actually work. So bats are very diverse. It's the second most diverse group of mammals, with more than 1,400 species distributed worldwide. You can find bats virtually everywhere, apart from Antarctica and some isolated islands in the Pacific, mostly, but even some species migrate close to the Arctic, not in the Arctic, but you find species close to the Arctic where they're able to hibernate. They have very different ecology. They have very different morphologies as well, adaptations for many different things. So they're a very interesting group of animals to study, simply because they can be easily sampled. You can find them anywhere, and they have a lot of different adaptations, which makes them interesting to study in many different contexts. And one of the things that it was interesting when they started studying the interactions of bats or diets of bats and many other different species as well, is that they have tried to recover the amount of DNA present in the diets of these species and try to see if this can actually, we can use this to represent abundance, for example. So can I actually measure the strength of an interaction based on the amount of DNA that I can find somewhere, for example? So if I find a species of Lepidoptera that has 1,000 sequence in the feces of a bat, can actually say that this Lepidoptera has been eaten 10 times more than another species where you have found just 100 sequences in the feces of the same bat. So can we make this comparison and use the amount of the DNA that we found from one species in a place to actually use as account data or to represent the abundance of this species in the environment or in that place? So this is a very tricky thing to do. There are many, there are systems where this works, other systems where this doesn't work, and you need to investigate this more or less case by case in order to be able to use this or not. But it's something very difficult to do, simply because many species, they have different also abilities to digest the DNA and the body parts of each species as well. So if you think about a bat, for example, digesting a beetle is probably not the same as the same bat digesting a lepidoptera or a butterfly, just because the butterfly has many more soft parts where they are easy digestible, and they are expected to not leave so many DNA behind. So there are many ecological aspects of how the digestion works and how these samples are actually recovered as well, that makes difficult many times for anyone to be able to use the amount of DNA present in a sample to actually characterize it as the strength of the interaction in many different contexts. So you can see here that depending on the way that actually you use this count data, you can use the raw sequence, the relative abundance. So you can use the percentage of that sequence in the DNA in order to describe the interactions, the strength of interaction. And if you use this, you can see there are big changes in terms of how you perceive the interactions in the network if you discard or if you use only more frequent DNA or less frequent DNA. So depending on also on how you address this question and the way that you count this data from the amount of DNA present on this environment, you can also reach very different answers, ecological answers in your study. And this is important to think about as well when you are considering about using the amount of DNA as a proxy for abundance in any ecological context. It also can change the perception of the interactions as well. So if you think about the relative read abundance, so the frequency of sequences in the sample, and you consider also the removal of some of the sequence in order to represent this abundance of the frequency of the sequences, you also find different network structures across your study as well. So it's very important to sit and closely think about what's the best way, considering your taxa that you're studying, but also the ecological context where you are retrieving this DNA. This is also very important because if you use as when you're doing the matrix of interactions to calculate to measure the structure of the network, if you use abundance data or frequency data or binary data, you can also reach different ecological conclusions in your study depending on the network metric they're using as well. So if you use just the presence of or absence of a species, for example, in an environment, to make a network of interactions, or if you use how much DNA is actually representing the abundance of this taxa, you're mainly forming networks that are very different in terms of this structure. So it's very important not only to think about this from the genetic side, but also from the network side. And there are many challenges as well, because we have a lot of sequences that are actually artificially created during the PCR procedures. So we need to think sometimes as well, not only about the amount of DNA that if we're going to use DNA to represent abundance, but also how much DNA should we be removing in terms of how many copies of sequence should we be removing in order to actually have only sequences that are real in our sample. So usually artificially created sequence in PCRs, they are more rare. So if you remove sequences that are very rare in your sample, you should be removing all the artificially created sequence. So this is one of the main strategies that people use to deal with these non-real species that could be detected in genetic samples. And this is also a way to avoid secondary predation as well. If you think about that species are not isolated in terms of how they interact, that one species could be eating another species, that could be eating another species. So if you only focus on answering a question based on these two species, first species interacting, but this second species also eating a third species, you can find within the diet of the first species also the DNA from this third species that has been eaten. So for example, you can find, it's possible to find the DNA of the fish that's purple in the diet of the DNA of the fish that's that is blue. So we have, when we remove this rare sequence, it's also a way to control for this this interaction that actually they don't happen in the wild, but you might be capturing them as just because you're sampling secondary predation. There are many different steps that you need to look at this in order to control and to understand how these bioinformatic approaches, they should have consequence for the structure of the network, but also there's the primers as well. So if you use different primers, you're probably likely to be sampling different species as well, because primers are good in sampling or in capturing the diversity, genetic diversity of different groups. So there's a study from Alberti from 2017 that he has shown that if you use different primers, you will also be describing different species within these networks or these genetic pools of DNA as well, which can also lead to different perceptions of the networks as well. So for example, here I have used the ZEO primers to describe the species present in the diets of the bats that I was studying. And you can clearly see here that there's a lot of pink dots that represent Lepidoptera. So most of the diets of the bats that I have sampled in these networks, they're actually Lepidoptera. So the bats would be eating Lepidoptera, but some of the things that have been proposed across the years, that the ZEO primers, they're good in describing Lepidoptera as well. So it's hard to know if the bats are actually eating more Lepidoptera in this case, so that's why you have more interactions and more species of Lepidoptera described in the diets of them, or if this is actually just a matter of the biases of the primers. So the primer is better in describing Lepidoptera, so we find more Lepidoptera in the diets of the bats, because we're using a primer that's good to do that as well. So we always need to be aware of these biases and these caveats in order to be able to understand how to approach and the networks, and also how to use this molecular data to build the networks. One interesting thing as well is that we have something that we call MOTU as well, so molecular operational taxonomic units, as I said before. So it's a way that you can pull sequences together that they are very similar in terms of the pairs of the base pairs that they actually have inside them, or in the order as well. So if two sequences are very similar, they should be pulled together if you use a high clustering threshold. So if I use, I'm calling a MOTU sequence that are 99% similar between them. So you end up having a lot of different sequences. But if you call a MOTU, or similar to what we say species, sequence that have 94% of similarity between them, you end up with less species in your sample as well. So this is also a approach that you need to think, anyone using this needs to think as well, because when you do that, you are increasing the number of species of your sample or decreasing them, and then making something that can be more artificial in terms of the number of species, or more real in terms of what you would find in nature as well. And this has a potential implication as well, or a relationship with how many copy numbers, how many sequences with a certain amount of copy numbers you will remove from your sample as well. So let's suppose that you remove from your genetic sample all DNA sequences that have only been found once, or twice, or 3 times, or 4 times, or 100 times. Once you start removing this rare sequence in order to control for the artificially created sequence during the process, you're also making the network more similar in terms of the structure between them as well, supposed to be. So we have made a study that we published last year with Dave has conducted the study. He's also going to be talking here in the course. So we have actually checked if we change this clustering thresholds or how we define a species or a motu in our network using a higher percentage of similarity between genetic samples or between the genetic sequence in order to call species, or a lower percentage, how this affects the network structure. So we have seen that some metrics such as nestedness and modularity, for example, they suffer a much less effect of clustering data together or dividing things into different species more easily, depending on the clustering threshold that we are using here. We have showed here the table which of the network metrics are also more resilient or more consistent in terms of the answer to these thresholds. And we have a case here where we have clearly applied this in order to answer this question. So we study here, this is a study that I have published from my PhD last year. So we're analyzing intraspecific variability in terms of the diets of species of bats, insectivorous bats in Costa Rica. So we have applied DNA barcode, meta-barcoding to describe the diet of these species. We used zero primers, so again, a lot of Lepidoptera in the diet of the species. And then we have looked at if different individuals, they have different positions in the network, but we have used the threshold. So we have called a species or a motu sequence that are 92% similar, as far as I remember well, like the percentage. But then you have the question: if I change the percentage of similarity that the sequence should have for me to describe that arthropod in the diet of this species, would my ecological answer change across this gradient? So we have applied different OTU thresholds, so different thresholds of similarity between genetic samples or DNA samples in order to call a species or an arthropod in the diet of these bats. And we have seen that even if we have changed this percentage of similarity, the answer that we have found is roughly the same. So that the ecological question that we are answering doesn't change even if we change some of the way that we analyze the data using DNA barcoding. We also have used Refraction curves. So if we used higher thresholds, so we are actually splitting species or motus in this case more easier because they have less similarity, more similarity between them. So more similar species would be called different species with a higher threshold. We would also be finding that the network they have more interactions as well. So the bats would be eating more things instead of when we are conservative and using a lower OTU threshold to describe the same interaction. So this is important because in this case, the ecological answer that we have found doesn't change very much across these thresholds, but in terms of the amount of species that the bats are eating or they're expected to be eating, there is a considerable change depending on the threshold and the parameters that we use in this analysis. I think that's it that I had to talk about today. I would like to thank for the Universidade Federal de Parana, the CAP Spring program, and also all the other places that I have worked at as well, and my friends and family for all the support across these years and my research as well. And I see you at the room for more questions, if anybody has any.